Welcome to this explainer. Okay, so if you are staring at the phrase Analytical Method Validation and literally feeling a cold sweat coming on, honestly, I completely get it. It sounds like an absolute compliance nightmare, doesn't it? Just a massive wall of acronyms and regulatory jargon. But don't worry, because today we are going to totally demystify the whole thing. We're going to break down these intimidating concepts into everyday, easy-to-understand terms. So by the end of this, you'll be speaking the language of validation like an absolute pro.
So, if you dive into the source material, you'll find this super formal definition. It basically says, validation is the process of establishing by laboratory studies that the performance characteristics of a procedure meet requirements for its intended use. Okay, legally, that giant mouthful just translates to one really simple question. Does this lab test actually do what we want it to do? Seriously, that's it. That is the entire heart of validation.
Here is our quick roadmap for today. 1. The rule makers. 2. The bullseye. 3. The VIP section. 4. The mathy stuff. 5. The toughness test. And finally, number 6, the big takeaway.
All right, kicking things off with Section 1, the rule makers. Who is asking for this?
To really understand why we spend so much time on this, we need to know who the bosses of the industry actually are. We're talking about massive global regulatory drivers that are pushing us to validate our methods. These are huge global bodies and guidelines like the ICH, the USP, and specific codes like CFR 211.165E from the FDA. And look, these aren't just polite suggestions. These guidelines explicitly demand that we establish and document things like accuracy, sensitivity, and specificity. Why? Because any analytical method intended for clinical samples absolutely has to be validated, just to ensure that the medicines we eventually take are actually safe.
Moving right along to Section 2, the bullseye, accuracy versus precision. People mix up accuracy and precision all the time, but they are actually super different. Accuracy is the closeness of agreement between the true value and the found value. Think of it like throwing a dart and hitting the exact dead center of the bullseye. Precision, on the other hand, is the degree of scatter among multiple tests. This is like throwing 5 darts and getting them all in the exact same spot. Now, even if that spot is way off in the corner, you are still highly precise, you know? You're just not accurate. In the lab, obviously we need our tests to be both.
And precision actually has layers. It breaks down a bit further into how we measure that degree of scatter. If we test it under the exact same conditions, like same day, same analyst, same machine, that's called repeatability. And then, if we test it under completely different conditions, maybe different days or entirely different labs, that is called reproducibility.
Now, that's all great for hitting the target in a perfectly pristine, controlled setting. But what if our sample isn't perfectly clean? What if it's like a totally chaotic, completely crowded, noisy room full of other stuff?
Which brings us to Section 3, the VIP section, specificity and selectivity.
Specificity and selectivity are officially defined as the ability to measure an analyte in the presence of interfering components. The easiest way to think about this, imagine your analytical method is an exclusive VIP bouncer at a club. The bouncer's only job is to walk into a massive, rowdy crowd of interfering junk, impurities, degradants, totally random molecules, and pluck out exactly the one specific VIP molecule we are looking for, completely ignoring everything else.
All right, on to Section 4, the mathy stuff, linearity, range, LOD, and LOQ.
I promise you, these mathematical limits are so much simpler than their acronyms make them sound. Let's look at LOD and LOQ. LOD is the limit of detection, which is just the lowest amount detectable. LOQ is the limit of quantitation, the lowest amount quantifiable with acceptable precision and accuracy. Think of it like this. LOD is like hearing a faint whisper at a really loud party. You know someone is there, you can detect them, but you have no clue what they're saying. LOQ is when they speak just loudly enough that you can actually hear the exact words and know the juicy gossip they are saying.
Then we have linearity, which is just a proportional response across a given concentration range. Let's make this super easy. If you double the amount of the drug in your sample, the test signal or response should exactly double too. It's just a perfectly straight line on a graph. And finally, for the math, we have range. This is the interval from the lower to the upper concentration that has suitable precision, accuracy, and linearity. Think of range as your method's Goldilocks window. It's the safe zone right between the lowest and highest amounts where our test still works perfectly and gives us results we can actually trust. Okay, so theoretically, our test is accurate, it's precise, specific, and mathematically sound. But what happens when the real world gets a little chaotic
and the lab gets a bit messy.
That brings us to Section 5, the toughness test, robustness versus ruggedness.
Now, these definitely sound similar, but they measure completely different types of toughness. Robustness, which is defined by the ICH, is the capacity to remain unaffected by small, deliberate parameter variations. Think of this like slightly tweaking a recipe in your own kitchen. Like maybe you accidentally bake a cake for 11 minutes instead of 10. Does the cake still turn out okay? If it does, your recipe is robust. Ruggedness, defined by the USP, is the reproducibility of results across entirely different labs, analysts, and instruments. This is like handing your favorite cake recipe to your friend in a totally different state, with a completely different oven. If they get the exact same cake, your method is rugged.
And finally, Section 6, the big takeaway. When we put it all together, look at that. You have successfully learned and categorized a dozen incredibly intimidating ICH and USP parameters. You know exactly how to hit the target, how to find your VIP molecule, the math limits, and just how tough the test really is. See, it's really all just everyday concepts applied to a laboratory setting.
So what is the bottom line here? Method validation establishes that our procedures consistently meet their intended purpose. And because of that, it has to be carefully planned out during method development. It is absolutely not an afterthought that you can just tack on at the end. And to keep all those rule makers happy, every single critical result, all your bullseyes, your VIP bouncer tests, your toughness checks, they must be completely and thoroughly documented.
The source material really hits the nail on the head when it says validation is essential but time-consuming for most analytical development laboratories. And yeah, for sure, it takes a massive amount of time and effort, but I want to leave you with this to think about. The very next time you open your medicine cabinet and safely take a pill, you literally have this exact rigorous process to thank. It kind of makes you wonder, what other invisible safety nets are just quietly protecting us every single day? Anyway, thanks so much for joining me on this explainer, and I really hope this complex topic finally makes perfect sense.